Every living cell runs like a small factory. To grow and divide, a cell must constantly produce proteins, increase in size, and move through the cell cycle. As long as this production line runs smoothly, the cell thrives. But when biosynthetic activity slows down, the rules that govern growth and division begin to change. What is not yet clear is how far this slowdown can go. Does a cell respond gradually to decreasing biosynthesis, or is there a critical point beyond which further reduction no longer matters? In this study, we set out to explore whether cells require a minimum level of biosynthetic activity to remain physiologically fit. We used budding yeast as a model system and designed an experimental framework that allowed us to reduce biosynthetic activity in two distinct ways. Firstly, we genetically deleted selected ribosomal protein genes such as RPL20A, RPL20B, and RPS6B, creating yeast strains with progressively lower protein synthesis capacities. Secondly, we applied a calorie restriction by limiting glucose availability, mimicking an environmental reduction in nutrient supply. This combination allowed us to observe how intrinsic biosynthetic limitations and external nutrient availability influence cellular behaviour. We found that when biosynthetic activity is reduced, cells grow more slowly, become smaller, and take longer to complete the cell cycle. This delay is not evenly distributed across all phases, but primarily affects the G1 phase, the critical stage where cells decide whether to commit to division. These observations suggest that cells closely monitor biosynthetic output before moving forward in the cell cycle. However, the most striking results emerged when we compared wild-type cells with ribosomal protein-deficient strains. In wild-type yeast, calorie restriction caused a clear decline in growth rate, cell size, protein content, metabolic activity, and glucose utilisation. The cells actively adjusted their physiology in response to reduced nutrient availability. In contrast, the strain lacking the RPL20A protein told a very different story. These cells already operated at a strongly reduced biosynthetic capacity under optimal conditions. When calorie restriction was applied, no further significant changes in growth or biosynthetic activity were observed. This led us to a central conclusion of our study. Cells require a minimum biosynthetic threshold to maintain physiological fitness. Once biosynthetic activity drops below this threshold, additional limitations, such as calorie restriction, have little measurable effect. Together, these findings reveal that the impact of calorie restriction is not universal, but is highly dependent on the cell's underlying biosynthetic state. By identifying biosynthetic capacity as a limiting factor for growth, cell cycle progression, and physiological efficiency, this work provides new insight into how genetic and environmental factors jointly define cellular function.